Hello everyone, welcome back to our Biology 2401 lecture series. I am Dr. E. Today we go over chapter 4, tissues, the living fabric. When we look at our body, we often think of organs like the heart, the lungs, or the skin. But have you ever wondered what actually makes up those organs? It all starts with the tissues, groups of similar cells working together for a common purpose. In this chapter, we will explore the four main tissue types and review how they repair after injury. So let's dive in and discover how tissues form the fabric that holds an entire body together. Before we dive into the details of chapter 4, it is important to ask, why do we study tissues in the first place? The answer is because tissues are the foundation of every organ in the body. When tissues change, for example, through injury, inflammation, or disease, the function of the entire organ changes too. So for anyone entering healthcare, this knowledge is essential. Nurses assess tissue damage in wounds and burns. Lab professionals analyze tissue samples for cancer. Imaging technicians interpret how healthy or injured tissues appear on scans. And doctors diagnose many conditions by understanding what's happening at the tissue level. So studying tissues isn't just academic. It's a skill that helps you recognize disease, understand patient conditions, and make better clinical decisions. Now let's dive into the world of tissues. Tissues are groups of similar cells in structure that perform a common or related function. The study of tissues is called histology. In the body, there are four basic types of tissues: epithelial tissues, connective tissues, muscle tissue, and nervous tissue. We will review each of the tissue types throughout this lecture. This figure provides an overview of the four basic tissue types. Epithelial tissue forms boundaries between different environments. It protects, secretes, absorbs, or filters substances. Epithelial tissue is found in the lining of the digestive tract organs and other hollow organs. It is also found in glands such as the pancreas and in the epidermis of the skin. Connective tissue supports, protects, and/or binds other tissues together. Connective tissue is found in bones, tendons, ligaments, fats, and other soft padding tissue. Muscle tissue contracts to cause movement. There are three types of muscle tissue. Skeletal muscle is attached to bones. Cardiac muscle is found in the heart. Smooth muscle is found in the walls of hollow organs, for example, the walls of the stomach. Nervous tissue provides internal communication throughout the body. Nervous tissue is found in the brain, spinal cord, and nerves. We will review each of the tissues in detail as we go through this lecture. To be able to view tissues under the microscope, tissues must be fixed, sectioned, and stained. Fixing preserves the tissue using a solvent. Sectioning cuts tissues into thin slices for light or electrons to be transmitted. And finally, staining enhances contrast. Light microscopy uses colored dyes as stains, while electron microscopy uses heavy metals. We start our review of each tissue type with epithelial tissue. Epithelial tissue cover body surfaces or line internal cavities. They can be divided into two forms: covering and lining epithelium and glandular epithelium. Covering epithelium forms the outer layer of the skin, lines open cavities, and covers walls of organs of the ventral cavity. Glandular epithelium forms glands of the body. Epithelial tissue function in protection, absorption, filtration, excretion, secretion, and sensory perception. Epithelial tissue have 5 distinguishing characteristics. Number 1 is polarity. Number 2 is specialized contacts. Number 3, they are supported by connective tissue. Number 4, they are avascular but highly innervated. And finally, they have a high regenerative capacity. We will review each characteristic over the next slides. The first characteristic is polarity. All epithelial tissues have 2 surfaces that differ in structure and function. These are the apical surface and the basal surface. The apical surface is not attached to surrounding tissue and is exposed to either the outside of the body or the cavity of an internal organ. Most apical surfaces are smooth, but some have specialized finger-like projections called microvilli. The basal surface is attached to the underlying connective tissue. The basal surface attaches to the basal lamina, an adhesive sheet that holds basal surfaces of epithelial cells to underlying cells. Epithelial cells are said to exhibit apical basal polarity. The next characteristic is specialized contacts. Except for glandular epithelium, epithelial cells fit closely together to form continuous sheets. The sides of adjacent cells are tied together by tight junctions and desmosomes. Recall from chapter 3 that tight junctions prevent substances from leaking through spaces between cells, while desmosomes keep cells from pulling apart. Epithelial tissue is supported by connective tissue. All epithelial sheets rest upon and are supported by connective tissue. Between the epithelia and connective tissue is a basement membrane. The basement membrane is made up of 2 layers, a basal lamina and a reticular lamina. The basement membrane reinforces the epithelial sheet, helps it resist stretching and tearing, and defines the epithelial boundary. Epithelial tissue is avascular but innervated. Avascular means it contains no blood vessels. Epithelial cells are nourished by substances diffusing from blood vessels in the underlying connective tissue. Although avascular, epithelial tissue is supplied by nerve fibers. Epithelial tissue has high regenerative capacity. Some are exposed to friction and their surface cells rub off. Others are damaged by hostile substances in the environment. When their apical basal polarity and lateral contacts are destroyed, epithelial tissue begin to reproduce themselves rapidly. Adequate nutrition is required for epithelial cells to replace lost cells by cell division. How are epithelial tissues classified? All epithelial tissues have 2 names. The first name indicates the number of cell layers and can be divided into simple or stratified epithelia. Simple epithelia consists of a single cell layer. Stratified epithelia consists of 2 or more cell layers. The second name indicates shape of cells. Shapes could be squamous, which means flattened and scale-like, cuboidal, which means box-like or cube-shaped, and columnar, which are tall or column-like cells. This figure shows classification based on the number of cell layers in an epithelium. In this classification, there are 2 types of epithelia, simple and stratified. A simple epithelium consists of 1 cell layer. A stratified epithelium consists of 2 or more cell layers stacked on each other. Regardless of how many cell layers an epithelium contains, it has 2 surfaces, an apical surface and a basal surface. This figure shows classification of epithelial tissue based on the shape of the cells. In this classification system, there are three types of epithelia: squamous, cuboidal, and columnar epithelia. Note that the shape of the cells influences the shape of the nucleus. In squamous epithelium, cells are flattened and scale-like, and the cell's nuclei are also flattened. In cuboidal epithelium, cells resemble cubes, approximately as tall as they are wide. The nuclei of cuboidal epithelial cells are spherical. In columnar epithelium, cells are tall and relatively thin. In other words, they are shaped like columns. The nuclei of columnar epithelial cells are also tall and thin, and usually located closer to the basal surface of the cell. Now, let's start our review of different types of epithelial tissue. We start with simple squamous epithelium. A simple squamous epithelium is the simplest of the epithelia. It has a single layer of flattened cells with disc-shaped central nuclei and sparse cytoplasm. In terms of function, they allow materials to pass by diffusion and filtration in sites where protection is not important. They also secrete lubricating substances in linings of ventral body cavities. They can be found in the kidney glomeruli, air sacs of the alveoli of the lungs, linings of the heart, blood vessels, and lymphatic vessels, as well as the serous membranes. The image here shows a simple squamous epithelium forming part of the alveolar walls of the lungs. The next is simple cuboidal epithelium. Simple cuboidal epithelium has a single layer of cube-like cells with large spherical central nuclei. They are involved in secretion and absorption. Simple cuboidal epithelium can be found in the kidney tubules, ducts, and secretory portion of small glands. Next is simple columnar epithelium. Simple columnar epithelium has a single layer of tall cells with round to oval nuclei. Some of the cells have microvilli, while some have cilia. Other layers may contain mucus-secreting unicellular glands called goblet cells. They are involved in absorption and secretion of mucus, enzymes, and other substances. Ciliated columnar epithelial cells move mucus or reproductive cells by ciliary action. In terms of location, non-ciliated columnar epithelium lines most of the digestive tract, from the stomach to the rectum. Non-ciliated columnar epithelium also lines the gallbladder and excretory ducts of some glands. Ciliated columnar epithelium lines the small bronchi, uterine tubes, and some regions of the uterus. Next, we review pseudostratified columnar epithelium. In pseudostratified columnar epithelium, cells vary in height and appear to be multi-layered and stratified. However, the tissue is in fact a single-layered simple epithelium. Pseudo means false, so the tissue appears stratified, although it is not. Some of the cells do not reach the free surface, and the nuclei are seen at different levels. Pseudostratified epithelium may contain cilia and mucus-secreting cells. It is involved in secretion of substances, particularly mucus and movement of mucus by ciliary action. It is located mostly in the upper respiratory tract, ducts of large glands, and tubules in the testes. Next is stratified squamous epithelium. As already mentioned, stratified epithelial tissues have two or more layers of cells. Stratified squamous epithelium is composed of several cell layers. The basal cells are cuboidal or columnar and are metabolically active, while surface cells are flattened and squamous. Stratified squamous epithelium can be keratinized or non-keratinized. In the keratinized type, the surface cells are full of keratin and are dead, while basal cells divide actively by mitosis and produce cells of the more superficial layers. Keratinized stratified squamous epithelium is found in the skin and functions to protect underlying tissues in areas subject to abrasion. Non-keratinized stratified squamous epithelium is found in moist areas like the esophagus, mouth, and vagina. The next is transitional epithelium. This type of epithelium resembles both stratified squamous and stratified cuboidal epithelium. The basal cells are cuboidal or columnar, while surface cells appear dome-shaped or squamous. Transitional epithelium is found in the bladder, ureter, and urethra. The epithelial cells can change their shape when stretched, allowing for increased urine flow. In the case of the bladder, stretching allows for more space for urine storage. Next, we will review glandular epithelium. Glandular epithelium forms glands of the body. A gland is one or more cells that make and secrete an aqueous fluid called a secretion. And they can be classified based on the site of product release and number of cells forming the gland. In terms of the site of product release, you can have endocrine or ductless glands that produce hormones and exocrine or glands that secrete their products through ducts. In terms of number of cells, you can have unicellular glands or multicellular glands. This figure shows formation of multicellular exocrine glands. Multicellular epithelial glands form by invagination or inward growth of an epithelial sheet. Cells from an epithelial sheet extend inwards to form an invaginated epithelial sheet. Exocrine glands retain the connecting cells, which forms a duct that transports secretions to the epithelial surface. Endocrine glands lose their ducts during development. They secrete hormones into the interstitial fluid. These hormones then enter the blood and are distributed throughout the body. Endocrine glands are ductless glands. Their secretions are not released into a duct. Instead, the secretions are released into the surrounding interstitial fluid, which is then picked up by the circulatory system. Endocrine glands release hormones by exocytosis. Hormones are chemical messengers that travel through lymph or blood to their specific target organs to produce a response. Target organs must have receptors for hormones. Exocrine glands secrete their products into ducts, and the secretions are released onto body surfaces such as the skin or into body cavities. Examples include mucous glands, sweat glands, oil glands, salivary glands. Exocrine glands can be classified as unicellular or multicellular. The only important unicellular glands are mucous cells and goblet cells. They are found in epithelial linings of intestinal and respiratory tracts. All unicellular exocrine glands produce mucin. Mucin is a sugar protein that can dissolve in water to form mucus. Mucus forms a slimy, protective, and lubricating coating on the surface of cells. Unlike unicellular exocrine glands, multicellular exocrine glands are structurally more complex. They consist of two parts, an epithelium-derived duct and a secretory unit. Multicellular exocrine glands can be classified based on their structure and mode of secretion. Structurally, multicellular exocrine glands are classified on the basis of their duct structure as either simple or compound. Simple glands have unbranched ducts. Compound glands have branched ducts. The glands are further categorized by their secretory unit as either tubular, alveolar, or tubulo-alveolar. Tubular glands form tubes, for example, intestinal and stomach glands. Alveolar glands form flask-like sacs, for example, sebaceous glands. Tubulo-alveolar glands have both tubular and alveolar secretory units, for example, salivary glands. Multicellular exocrine glands secrete their products in different ways, and so can be classified functionally as merocrine, holocrine, and apocrine. Merocrine glands are the most common and secrete their products by exocytosis as the secretions are produced. Secretion does not alter secretory cells. Examples include sweat glands, pancreas, and salivary glands. In holocrine secretion, secretory cells accumulate products within until the entire cell ruptures. The cells are replaced by division of underlying cells. An example is sebaceous glands. Apocrine glands accumulate product within, but only the apex of the cell ruptures. The cell repairs the damage and repeats the process. There is some controversy if apocrine glands are present in humans. Mammary glands are the closest example, but most scientists classify mammary glands as merocrine glands. The next major tissue type is connective tissue. Connective tissue is the most abundant and widely distributed primary tissue in the body. It functions in binding and support, protecting, insulating, storing reserve fuels, transporting substances within the body. There are four main classes of connective tissues: connective tissue proper, cartilage, bone, and blood. We will review each of them as we go. Two common characteristics make connective tissues different from other tissues. The first is extracellular matrix of non-dividing portion of connective tissue, in which living cells are suspended. The second is common origin. All connective tissue arise from the mesenchyme during embryonic development. All connective tissues are made up of three components: ground substance, fibers, and cells. Ground substance and fibers together make up the extracellular matrix. Composition and arrangement of all three elements varies from one connective tissue to another. Ground substance is unstructured gel-like material that fills spaces between cells. It is the medium through which solutes diffuse between blood capillaries and the cells. Ground substance is composed of interstitial fluid, cell adhesion proteins, and proteoglycans. Cell adhesion protein form the glue for attachment. Proteoglycans consist of a protein core with attached large polysaccharides called glycosaminoglycans. For example, chondroitin sulfate and hyaluronic acid. There are three types of connective tissue fibers. These are collagen fibers, elastic fibers, and reticular fibers. Connective tissue fibers provide support. Collagen fibers are the strongest and most abundant type. They are tough and provide high tensile strength. Elastic fibers are a network of long, thin elastin fibers that allow for stretch and recoil. Reticular fibers are short, fine, and highly branched collagenous fibers. They have a different chemistry and form from collagen fibers. Now let's review connective tissue cells. There are two main types of cells: immature and mature cell types. Immature cells are called blast cells, and they typically secrete ground substance and the matrix fibers. Site cells are mature cells, and their role is to maintain the matrix. Examples: fibroblast in connective tissue proper becomes fibrocytes. Chondroblast in cartilage become chondrocytes. Osteoblast in bone tissue become osteocytes. Other cell types in connective tissues include adipocytes, leukocytes or white blood cells, mast cells, and macrophages. Adipocytes store energy as fat. Leukocytes respond to tissue injury. Examples of leukocytes include neutrophils, eosinophils, and lymphocytes. Mast cells detect foreign microorganisms and initiate local inflammatory responses. They contain secretory granules with chemicals to mediate inflammation. Some of the chemicals include heparin and histamine. Macrophages break down foreign material and dead cells. Macrophages can be fixed on connective tissues or migrate freely throughout the matrix. This diagram depicts the components of connective tissue and their arrangement. The major components are fibers, ground substance, and cells. Fibers and ground substance make up the extracellular matrix. The three major components of the model are summarized as follows. Fibers. A network of three intertwined fibers include, from the thickest to the thinnest, collagen, elastic fibers, and reticular fibers. Ground substance fills the space around the fibers and cells. Cells are clustered around the fibers and capillaries. They include fat cells, mast cells, fibroblasts, and macrophages. This is an overview of the types of connective tissue. Connective tissue consists of four main categories: connective tissue proper, cartilage, bone, and blood. Connective tissue proper contains two subcategories: loose connective tissue and dense connective tissue. Loose connective tissue is subdivided into three subcategories: areolar, adipose, and reticular connective tissue. Dense connective tissue is subdivided into three: regular, irregular, and elastic connective tissue. Cartilage is divided into three subcategories: hyaline cartilage, elastic cartilage, and fibrocartilage. There are no subcategories for bone and blood. We start our review of connective tissue types with areolar connective tissue. It is the most widely distributed connective tissue and serves as the universal packaging material between other tissues. It binds body parts together, allowing them to move freely over each other. Areolar connective tissue has a gel-like matrix containing all three fiber types. The cells include fibroblasts, macrophages, mast cells, and some white blood cells. In terms of function, areolar connective tissue wraps around and cushions organs. It also plays an important role in immune responses through the activity of white blood cells and macrophages. Areolar connective tissue is widely distributed under epithelial of the body. For example, it forms the lamina propria of mucous membranes, packages organs, and surrounds capillaries. Next is adipose tissue. Adipose tissue is very similar to areolar connective tissue, but with very sparse matrix. Adipose tissue has densely packed fat cells called adipocytes. Large fat droplets within the cell push the nucleus to the side. The functions of adipose tissue include to provide reserve fuel, insulate against heat loss, support and protect organs. It is found under the skin in sebaceous tissue, around kidneys and eyeballs, and within the abdomen and breast. Next is reticular connective tissue. Reticular connective tissue resembles areolar tissue, but its fibers are thinner reticular fibers. It has a loose network of reticular fibers in a gel-like round substance. The fibers form a soft internal skeleton or stroma that supports other cell types, including white blood cells, mast cells, and macrophages. It is found in lymphoid organs, including lymph nodes, bone marrow, and spleen. Next is dense regular connective tissue. It is primarily composed of closely packed bundles of parallel collagen fibers with a few elastic fibers. The measured cell type is fibroblast. Dense regular connective tissue attaches muscles to bones or to muscles. It also attaches bones to bones at joints. It can withstand great tensile strength when pulling forces are applied in one direction. Dense regular connective tissue is found in tendons and ligaments. The next is dense irregular connective tissue. It has the same elements as dense regular connective tissue, but bundles of collagen are thicker and irregularly arranged with some elastic fibers. Fibroblast is the measure cell type. Its role is to withstand tension exerted in many directions and provide structural support. It is found in fibrous capsules of organs and joints, dermis of the skin, and submucosa of digestive tract. Dense elastic connective tissue. This is basically dense regular connective tissue containing a high proportion of elastic fibers. Its main function is to allow tissue to recoil after stretching. It also maintains pulsatile flow of blood through arteries and aids passive recoil of lungs following inspiration. It is found in walls of large arteries, within certain ligaments associated with the vertebral column, and within the walls of bronchial tubes. The next major connective tissue type is cartilage. The matrix of cartilage is secreted by chondroblasts, the immature cells. Meanwhile, chondrocytes or mature cells maintain the matrix. Chondrocytes reside within cavities in cartilage called lacunae. Cartilage is avascular but receives nutrients from surrounding membrane or the perichondrium. There are three types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage is the most abundant and appears as shiny bluish glass. It has an amorphous but firm matrix with collagen fibers forming an imperceptible network. Chondroblasts produce the matrix, and when mature, as chondrocytes, lie in cavities called lacunae. Chondrocytes account for only 1 to 10% of the cartilage volume. Hyaline cartilage provides firm support and reinforcement. It serves as resilient cushion and resists compressive stress. Hyaline cartilage forms most of the embryonic skeleton before bones form, and it covers the ends of long bones in joint cavities. Hyaline cartilage forms costal cartilages of the ribs, cartilages of the nose, trachea, and larynx. Elastic cartilage is similar to hyaline cartilage, with more elastic fibers in its matrix. Its role is to maintain the shape of a structure while allowing great flexibility. It is found in the ear and epiglottis. Fibrocartilage is between hyaline cartilage and dense regular connective tissue. Its matrix is similar to but less firm than that in hyaline cartilage. It has predominantly thick collagen fibers. It's found in areas such as the intervertebral disc, pubis symphysis, and meniscus of the knee, where its tensile strength allows it to absorb compressive shock. The next major type of connective tissue is bone or osseous tissue. Bone has a hard calcified matrix containing many collagen fibers. Immature bone cells called osteoblasts produce the bone matrix, while osteocytes lie in lacunae and maintain the matrix. Bone is richly vascularized. Bone supports and protects body structures. For example, the skull encloses and protects the brain. Also, bone provides levers for the muscle to act on. Bone stores calcium, fats, and other minerals. The red bone marrow inside bones is a site for blood cell formation or hematopoiesis. The next type of connective tissue is blood. Blood is the most unusual type of connective tissue because it is a fluid. Blood consists of blood cells surrounded by a fluid matrix called blood plasma. The blood cells include red blood cells, white blood cells, and platelets. Red blood cells are the most abundant. Blood functions in transport of respiratory gases, nutrients, waste, and other substances. The next major tissue type is muscle tissue. Muscle tissues are highly vascularized, and they are responsible for most types of movement. Muscle cells possess myofilaments made up of actin and myosin proteins that bring about contraction. There are three types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscles are long, cylindrical, and have multiple nuclei. They appear banded or striated. They are also called voluntary muscle because they can be consciously controlled. Skeletal muscles are attached to bones or occasionally to skin, where they bring about voluntary movement. Cardiac muscles are found only in the heart, and they are involuntary. Like skeletal muscles, cardiac muscle contains striations, but their cells have only one nucleus. Cells of cardiac muscles can have many branches that join branches of other cells at special junctions called the intercalated disc. Contraction of cardiac muscle propels blood into the general circulation. Smooth muscles have spindle-shaped cells with one central nucleus. Smooth muscles have no visible striations and are under involuntary control. They are mostly found in the walls of hollow organs, where they function in propelling substances such as foodstuff along internal passageways. The next major tissue type is nervous tissue. Nervous tissue is the main component of the nervous system, which regulates and controls body functions. The nervous system is made up of two specialized cells, namely neurons and supporting cells, also called neuroglia. Neurons respond to stimuli through processes called dendrites and transmit electrical signals along processes called axons. Supporting cells support, insulate, and protect neurons. Now we will review membranes. Membranes are organs of more than one tissue that cover and line structures. There are three types of coverings and lining membranes: cutaneous membranes, mucous membranes, and serous membranes. Cutaneous membrane is another name for the skin. Cutaneous membrane is made up of keratinized stratified squamous epithelium, or the epidermis, attached to a thick layer of connective tissue, or the dermis. Unlike other membranes, the skin is a dry membrane. We will review the skin in detail in the next chapter. Mucous membranes, also called mucosae, line body cavities that open to the exterior, including hollow organs of the digestive, respiratory, and urogenital tracts. In all cases, they are wet or moist membranes covered by secretions or urine in the case of the urinary mucosa. Most mucosae contain either stratified squamous or simple columnar epithelium that lies over a layer of loose connective tissue called the lamina propria. Although mucous membranes secrete mucus, this is not a requirement. For example, digestive and respiratory mucosae secrete mucus, while the urinary mucosae does not. Serous membranes, also called serosae, are moist membranes of closed ventral body cavities. They are made from simple squamous epithelium resting on a thin areolar connective tissue. Recall from chapter 1 that serous membranes have visceral and parietal layers separated by serous fluid. Parietal serosae line internal body cavity walls, while visceral serosae cover internal organs. The serosae are named according to their location and specific organ association. The pleurae line the thoracic walls and cover the lungs. The pericardium encloses the heart. The peritoneum encloses the abdominopelvic organs. The next topic is tissue repair. When the body's barriers are compromised, the inflammatory and immune responses are activated for tissue repair. Repair starts very quickly, and tissue repair is a function of the inflammatory response. In fact, inflammation is a first step in tissue repair. Tissue repair can take place in two major ways: regeneration and fibrosis. In regeneration, the same kind of tissue replaces destroyed tissue, so original function is restored. In fibrosis, connective tissue replaces destroyed tissue, and the original function is lost. Now let's review the steps in tissue repair in a non-extensive skin wound. In stage 1, inflammation sets the stage. Trauma causes injured tissue cells, mast cells, and others to release inflammatory chemicals. Inflammatory chemicals make local blood vessels leaky, allowing white blood cells, fluid, clotting proteins, and other plasma proteins to seep into the injured area. Clotting seals off the injured area and prevents bacteria, toxins, and other harmful substances from spreading. The surface of the clot exposed to the air dries and forms a scab. In stage 2, organization restores the blood supply. The clot is replaced by the ingrowth of fragile capillaries that are characteristic of granulation tissue. This restores the vascular supply. Fibroblasts multiply and produce growth factors and collagen fibers that bridge the gap. When healing is complete, these fibroblasts revert to the resting stage or undergo apoptosis or programmed cell death. Macrophages phagocytize dead and dying cells and other debris. Surface epithelial cells multiply and begin to migrate over the granulation tissue. In stage 3, regeneration and fibrosis effect permanent repair. The fibrosed tissue matures and contracts, pulling the margins of the wound together. As it regenerates, the epithelium thickens under the scar, which detaches. A fully regenerated epithelium with an underlying area of scar tissue results. The scar may be visible as white lines depending on the severity of the wound. Let's review the regenerative capacity of different tissues. Regenerative capacity varies widely by tissue type, with some having high capacity for repair and others having limited regeneration. Tissues with high regenerative capacity include epithelial tissue, bone, areolar connective tissue, dense irregular connective tissue, and blood-forming tissue. Tissue with moderate regenerative capacity include smooth muscles and dense regular connective tissue. Tissues with virtually no regenerative capacity include cardiac muscle and nervous tissue of brain and spinal cord. That brings us to the end of chapter 4. Thank you for sticking around. Please stay tuned for chapter 5, the integumentary system, coming out soon. Bye for now.